Welcome to the last mini lecture on the first module, which is Introduction to AI and Machine Learning, and this is about applications. We are going to review some machine learning applications in the biomedical field, focusing on different data types and the type of task that can be performed, the models that can be applied to them, and some of the challenges. Let's start with the nucleotide sequence. FASTA is on our left here. FASTA is a very common format for nucleotide and protein sequences. It consists of a header, like in this case, preceded by a greater than sign, and then followed by the sequence in a new line. The sequence is in one-letter code. Some examples of tasks that machine learning could help using nucleotide sequence as inputs are DNA accessibility, the 3D genome organization, predicting enhancer-promoter interactions. As we saw in the previous lecture, recurrent neural networks are suited to deal with sequence dependencies like DNA sequences. So this would be one of the methods that you will find here as recommended model for this, but there are others. Some challenges that nucleotide sequence pose on machine learning is that they may contain repetitive regions, and repetitive regions can dominate the input, leading to overrepresentation of certain patterns. Thus, model may overfit to these patterns rather than learning generalizable features. Also, the genomic sequence can vary widely in length, so sometimes we have very long sequences or very short, like motif for transcription binding sites. And the longer the sequence, the higher the computational cost. Also, some algorithms require some fixed length as inputs, and this sequence then, if they are too long, they need to be pre-processed and normalized. Similar to nucleotide sequence, a standard format for protein sequence is FASTA, again, greater than, the header, and then the new line with a one-letter code. In this case, the letters represent amino acid. This data type can be used, for example, for protein structure prediction, like AlphaFold, given a sequence, predict the corresponding protein 3D structure. Also prediction of function or prediction of protein-protein interactions based on sequence. These are two of many other examples. There are a number of models recommended. Some of these we may review in future modules. One main challenge in this area is data leakage due to the information coming from homologous proteins. Data leakage occurs when information from the test set is inadvertently included in the training set, allowing the model to cheat by learning patterns that it would not have access to in real-world scenarios. So data sets like UniProt, which contains, that's where this sequence is coming from, or PFAM, which is for domain sequences, may include annotations that are propagated across similar sequences. If these annotations are directly or indirectly shared between training and test set, they introduce leakage. Another data type is 3D structure. As opposed to the previous one, in this case, the input is not a sequence, but the atomic coordinates, the structure. Machine learning is used for the 3D model refinement, which involves improving the initial 3D protein structure model to make it more accurate and closer to the native conformation. Also for quality assessment of the model and prediction of change in stability of the structure if a mutation is introduced. Like if we change, in the case of a protein, if we change an amino acid, for example, here in the structure, how that affects the stability of this structure. Listed here are some of the methods that are used. For example, graph computational networks, here shown as GCNs, using molecular graph, in this case, is very powerful at capturing long-range interaction, which is needed in the 3D structure. In this case, the amino acid in this helix, blue helix here, is far from probably an amino acid in this loop, green loop, and in the sequence-wise. but they are very close in structure. So these methods are good at capturing these interactions. Some of the challenges involved in this case is lack of data. If we compare the number of sequence data that we have in comparison to structure, is really very, very tiny. And especially if we are looking for protein complexes. There's also a lack of data for a special type of proteins which are disordered, basically. They do not have a structure that we can capture properly. Another data type we could encounter is gene expression data. The type of task is to predict, for example, co-expression. The input is normally genes and their expression levels. Of all these models I'm going to mention about clustering plays a pivotal role in analyzing gene expression data by grouping the genes, samples, or conditions with similar expression patterns. This is an unsupervised machine learning technique as opposed to the ones that we've been reviewing before. It provides insight into biological processes, regulatory mechanisms, and disease states, depending what we are studying. Challenges in this area when making connection to biology. Co-expression is not a synonym of functional connection. And also another thing is that the data has high dimensionality and noise. The next type is about mass spectrometry. In this case, the input is the mass spectra, which looks like the one on the left here. We have peaks with masses and some intensity, and detecting, for example, one of the prediction tasks or machine learning can help in detecting peaks, because this is critical as peaks corresponds to molecules, ions, or fragments of interest. An accurate detection influences downstream analysis such as compound identification, quantification, or biomarker discovery. So machine learning can be used for this task of peak detection. In this case, a convolutional neural network can process the raw 2D mass spectra or the images of the spectra to detect and segment the peaks. Some of the challenges in mass spectrometry involves the lack of standardized benchmark, that is, having high quality data sets to compare data with. Images like biomedical images, the ones that we have here on the left, or cryon electron microscopy, for example, for protein or other macromolecule structures, are very popular data types. Machine learning can be used to identify anomalies in medical images like a tumor. Convolutional neural networks is a popular method used with images. Some of the challenges here involve systematic difference in data collection, included but not limited to variations in imaging devices, the type of machines, the difference in the resolution, sensitivity, and calibration between devices, in the protocols for data acquisition, and so on. This systematic difference introduces bias and reduces generalization of the model. There's also the issue of hard to obtain large data sets of consistent data in some cases. Another data type is molecular structure. There are many formats for chemical structures. The 2D structure, like the one represented on the left for 5'-deoxyadenosine, and there's also a text version representation, for example, smiles. And these prediction tasks related to molecule are very, very diverse, but includes, for example, predicting the activity or toxicity of a compound, using the compound to dock into a protein, like a binding site, to see if it could bind. For structures, 2D structure or 3D structure, methods like the ones for 3D protein structure, CNN with molecular graph, are used. In molecular graphs, atoms are the nodes and the bonds are the edges, allowing this method to directly model the structure of chemical compounds. While a different method called recurrent neural network is used for the corresponding text representation of the compound, such as smiles, which is one of the used formats for text representations of chemicals. Actually, it is a sequential representation, making it suitable for the sequence-based models like the one I just mentioned, the recurrent neural networks that can learn patterns in the order of characters. Some challenges here is that limited availability of the experimental data for compounds because of the wide variety of possible molecules. The last data type we will review is protein-protein interaction networks. The input is the graph with proteins as nodes and edges indicate their interaction. Prediction tasks include polypharmacology side effects. This refers to the unintended biological effect or adverse outcomes caused by a drug interacted with multiple proteins or pathways rather than just the intended therapeutic target. One could simulate how a drug's binding to specific protein propagates through the BPI network, highlighting, for example, the patterns or proteins associated with the side effects. Methods like the graph embeddings are widely used with networks. Challenges had to do with the lack of environmental input, like, for example, knowing that certain proteins are located in particular compartments, and also with data completeness. Interaction networks can be incomplete. Here are the reference for this section, and hopefully you get a glance of some of the applications. Thank you.